The majority of neuroendocrine cancers occur sporadically. This means there is no underlying genetic cause. However, there are some neuroendocrine cancers that are linked to genetic disorders and inherited conditions. To understand how this happens, we need to look inside the cells of the body. Inside our cells are chromosomes made up of DNA. DNA is the cell's instruction manual. Genes are made of lengths of nucleotides, the repeating units found in DNA that guide our development and traits such as eye colour. Sometimes, if there are mistakes in the DNA, it can lead to genetic changes. These can occur randomly or be inherited. There are a number of different gene changes or faults that can lead to an increased risk of neuroendocrine cancers, also known as neuroendocrine tumours or NETs. The neuroendocrine gene faults are usually inherited in a dominant pattern, meaning that only one copy of the faulty gene is needed to cause the genetic condition. Therefore, each child of an affected parent has a 50% chance of inheriting the faulty gene. Families with a known history of these genetic disorders are encouraged to discuss genetic testing. It is important to understand genetic conditions and disorders associated with NETs because some genetic disorders or inherited conditions mean a person is at an increased risk of developing one or more types of NETs. Knowledge that a person or family carries a genetic risk of NETs empowers them to undergo screening tests. Screening can lead to earlier diagnosis and treatment of NETs. Multiple Endocrine Neoplasia, known as MEN, is a group of inherited benign or cancerous tumours affecting the body's network of hormone-producing glands. There are several types of MEN. The MEN1 gene is a tumour protection gene. When faulty, tumours can develop. MEN1 is associated most commonly with NETs of the pituitary and pancreas. MEN2 is when there is a faulty RET gene. The RET gene is involved in thyroid, parathyroid, and adrenal gland cell growth. NETs associated with MEN2 mutations are pheochromocytoma, also known as pheo, and medullary thyroid cancer, MTC. People with MEN1 and MEN2 mutations can have high calcium from an overactive parathyroid gland. Familial pheos and paraganglioma syndrome is the term used when there is a fault with the succinate dehydrogenase gene, commonly known as SDHA, SDHB, SDHC, or SDHD. These genes are responsible for suppressing tumour development. A fault in one of these genes can mean an increased risk of developing pheos, paragangliomas, kidney cancers, pituitary NETs, and gastrointestinal stromal tumours, GIST. Von Hippel-Lindau disease is caused by a fault in the VHL gene. The VHL gene helps to control blood vessel growth. People with VHL disease have an increased risk of developing benign tumours, kidney renal tumours, pheos, gastrointestinal and pancreatic NETs, central nervous system lesions, retinal hemangioblastomas, and inner ear tumours. Neurofibromatosis type 1 is caused by a fault in a tumour suppressor gene called NF1. Neurofibromatosis type 1 is associated with pheos, gastrointestinal, and pancreatic NETs. Genetic testing may not be necessary, as clinical signs can assist with diagnosis. Tuberous sclerosis is caused by a fault in the genes TSC1, TSC2, which are involved in regulation of cell growth. Tuberous sclerosis can cause benign tumours and a higher risk of developing pancreatic or insulinoma NETs. People diagnosed with NETs should ask their specialist whether their NET may have a genetic link, especially if they have a family history of other NETs or other cancers. A detailed family cancer history should be done. Genetic counselling should be done before genetic testing. For example, people diagnosed with pheo, paraganglioma, MTC. understanding and information about NETs and genetics is constantly evolving. It is vital to stay informed and see if any more recommendations have been made or discovered through research. Contact the NET nurse for further information and resources.